So the purpose of the electron transport chain. The purpose of the electron transport chain is to take those hydrogen ions that have been carried by the NADH and the FADH2 and push those into this intermembrane space. Notice as this is reduced, pardon me, that's oxidized. Okay. As this is oxidized, as this hydrogen then is pumped into this intermembrane space. Okay. And every step along the way of that electron transport chain, we're pushing hydrogen ions into this intermembrane space. So what's going to happen is we're going to end up with a very necessary electrochemical gradient. So glycolysis, Krebs cycle, electron transport chain. The electron transport chain produces our electrochemical gradient, and it's the electrochemical gradient across that intermitochondrial membrane that's going to be used to produce a ton of ATP. Okay. So where we have high ion concentration versus low ion concentration, it's just simple diffusion. Okay. Simple diffusion. That high ion concentration has to flow. And it has to flow, in this particular case, through one embedded enzyme. That enzyme is referred to as ATP synthase. ATP synthase is a tiny little molecular motor. It's truly a physical movement process that causes these things to happen. As the hydrogen ions flow down that electrochemical gradient, okay, we're going to have what's referred to as the proton motive force. So here's our hydrogens now. Okay. And they are able to then move down the ATP synthase, which is also embedded in this intermitochondrial membrane. And as they move, it's the only place they have to go. So they want to flow. They have to flow through the ATP synthase. They turn the motor that's on the bottom of this, and referred to as the proton motive force. That motion of turning the ATP synthase opens up energy that's then used to phosphorylate ADP into more ATP. So each hydrogen binds physically with that spinning rotor on the bottom of the ATP synthase. Each hydrogen ion makes a complete revolution, and the turning of that rod at the bottom of the ATP synthase, okay, physically opens up catalytic sites active that allow for the phosphorylation of ADP. So much so that we can produce as much as 28 ATP. So here we have our inner mitochondrial membrane, our inner mitochondrial matrix. Okay, we have from our electron transport, we have a high electrochemical gradient of hydrogen ions. They have to flow. They're going to flow down through this ATP synthase. That proton motive force moves this rotor at the catalytic head rotor of the ATP synthase. And as that happens, it makes available the different enzymes that now can cause the phosphorylation of this ADP. And we've got a lot of these hydrogen ions. We're moving a lot, and we're spinning this rotor, and we can make up to 28 ATP. Now, with 28 ATP, we can power a multicellular organism. We can power a eukaryotic cell. Okay? Chemiosmosis, proton motive force, making a bunch of ATP.